My dear students, welcome to my today's online class on one more topic in paper 6 of semester 5 of zoology subject, that is genetics and biotechnology, prescribed by Bangalore City University. And today we are discussing about session 23, that is pertaining to physical, chemical and biological mutagens. In my previous session, I have taught you about the concept of mutation, types of mutation, and I have just briefed you about the concept of mutagen, an agent which brings about mutation. In this class, we are going to learn in detail about mutagens, different types of mutagens like physical, chemical and biological, and their effects. Okay? And I am Dr. MEERA B K, Associate Professor, Department of Zoology, Maharani Science College for Women, Bangalore 1. Before starting the session, I would like to thank my good friend and colleague, Dr. Lata V. Madam, who has prepared these quality PPTs, very informative PPTs pertaining to the concerned topic. And my role is just to give the voice to these quality PPTs. Thank you, Madam, for your timely help. We shall start the session. Now, here you can see a plate. So this topic belongs to module 2 or unit 2 and is about physical, chemical and biological mutagens. You can see here the UV radiations and X-rays, which do serve as mutagens, okay, under radiations. And many chemicals do serve as mutagens. For example, cigarette smoking, and then nitrate and nitrate preservatives, and then barbecuing, then benzyl peroxide, and many number of chemicals serve as potent mutagens, bringing about harmful mutations. Then next category is biological mutagens. Most of them are infectious agents, right, like viruses, bacteria, and one more category, what we call transposons, or popularly called jumping genes. So human papillomavirus HPV that causes cervical cancer, and then Helicobacter pylori, which causes ulcers in the intestine. So these are all different types of mutagens which bring about mutations, that is changes in DNA material. One by one we shall study in detail. So you will learn about different agents of mutation, and you will also try to understand the actions of these mutagens in their surroundings. Okay? Then, the contents of this talk has been divided into 4 subheadings: definition of mutagens, then details of physical mutagens, then chemical mutagens and biological mutagens, followed by assessment questions, of course. Now, what is the definition? One more question normally asked. Mutagens, muta means mutation, gen means to produce. Any agent that is responsible for induction of mutation is called mutagen. These are the agents that bring about mutation Three different categories of common mutagens are observed in nature. They are physical agents, chemical agents, and biological agents. And here is a schematic representation to brief you about the details of my talk in a nutshell. Alright? Then. So again, this is a plate briefing you about different types of mutagens. Basically, you find physical mutagens, chemical mutagens, and biological mutagens. Under physical mutagens, you find ionizing radiations and non-ionizing radiations. Then under ionizing, you find particulate matter and non-particulate matter. Under particulate matter like alpha rays, beta rays, neutrons, cobalt, cesium, radioactive cobalt, radioactive cesium. These are all mutagens coming under particulated matter of ionizing radiations. Under non-ionizing radiations, you find gamma rays and X-rays. Then coming to chemical mutagens, you find base analogues, chemicals, intercalations, alkylating agents. And each category of this will be taught in detail with suitable examples. And then the third category of mutagens happen to be the biological mutagens. They are viruses, bacteria, and transposons. We shall study them in detail one by one. And then, based on their effect, mutagens can also be classified in yet another way as teratogens, carcinogens, clastrogens, non-specific category. Teratogens are the mutagens which bring about congenital malformations. Carcinogens are the mutagens which induce tumor formation. They are cancer-causing agents. Clastrogens are the ones which bring about chromosomal abnormalities. And non-specific category, in general, they bring about DNA damage. So, based on the effect, mutagens are also classified into teratogens, carcinogens, clastrogens, and non-specific category of mutagens. Now, let us talk about physical mutagens and that radiations. The radiations are the first mutagenic agent which were reported as early as in 1920. UV rays, ultraviolet rays, X-rays, alpha rays, neutrons, and other ionizing radiations, and also non-ionizing radiations come under this category of physical mutagens. So the radiation effects at a molecular level do damage the genetic material, that is DNA, or the nucleotide structure. And the damage can be either lethal, having detrimental effects, or sublethal, tolerable effects. Then apart from this, the electromagnetic radiation is also one of the known mutagens that cause either lethal or sublethal mutations. And one of the common type of radiation, the ionizing radiation, produces the free radicals that damage not even the DNA but also the proteins and lipids present in a cell, bringing about mutation. Alright? Then, so the rapidly dividing cell types are one of the target types. Target tissue cells for ionizing radiations like X-rays to cause mutation. However, the severity of the damage depends upon the dose of radiation that is given, okay, to which particular dose the cell is exposed is of paramount importance in deciding the severity of damage, and it is directly proportional, of course. Higher is the dosage, more is the severity, and vice versa. So this radiation causes cross-linking of DNA or protein, then chromosomal break, strand break, or loss of chromosomes. And also at molecular level, it causes deletion of nitrogenous bases or DNA strand breakages. Whatever be the cause, whatever be the happening because of exposure to radiations, ultimate result is the mutation. Alright. Then let us talk about X-rays, which are the most common type of ionizing radiation used in many of the medical practices for various diagnostic purposes. At the molecular level, the lethal dose of X-ray, 350 to 500 rems, it breaks the phosphodiester bonds between the DNA and therefore results in the breakage of the strands. Then it causes multiple strand breakage and results in the deletion of a portion of the DNA. And if the strand breakage occurs in both the strands, it will become lethal to the cell. Alright. Then UV rays. The ultraviolet rays belong to non-ionizing type of radiation. Okay. And they are absorbed by DNA as well as protein at 260 nanometers and 280 nanometers respectively. Alright. And the major causes of UV radiation are base deletion, deletion of nitrogenous base, breakage of the strand, cross-linking, and generation of nucleotide dimers. And this UV light can be classified into 3 different categories based on the nanometer range. UVA, ultraviolet radiations, A, nearly visible range, 320 nm. This causes the formation of pyrimidine dimers. By now you know nitrogenous base pairs are of 2 types, purines and pyrimidines. And this UVA category causes pyrimidine dimers. And then UVB, next category, 290 to 320 nm. It is emitted by sunlight and it is one of the major mutagens that causes in DNA that changes DNA, and this type is highly lethal. Okay. Then UVC, that is range is 180 to 290 nm. One of the most energy consuming form of UV, and it is extremely lethal. So whatever be the category, all the 3 are mutagenic in nature. And the UV induced mutations are normally dimer formation like thymine-thymine dimer or thymine-cytosine dimer, which are commonly formed as these lesions block replication of DNA as well as transcription. So if not repaired, they cause serious health effects. Here you can see the normal DNA, the first diagram. When it is exposed to UV radiations, there is distortion of DNA structure, and you can see in the third diagram where there is TT dimer formation, thymine-thymine dimer formation. Okay. And the normalcy can be brought back by DNA repair mechanisms. If it is not done, that will lead to serious health issues. Then heat. Heat is another important mutagen that causes mutation in the DNA In fact, when we heat the DNA above 95 degrees Celsius, the DNA becomes denatured in the sense the double-stranded DNA, undergoes denaturation, and you will get single-stranded DNA, right? The hydrogen bonds will be broken down. Also, extreme heat damages DNA and breaks the phosphodiester bonds also. Then, after having discussed about physical mutagens, we shall talk about chemical mutagens. Chemical mutagens are the chemicals that induce mutations. They are actually dangerous, and this mutagenic effect of the nitrogen mustard was reported in 1942, as early as 1942 by Charlotte Orbeck. And what are the chemical agents that induce mutation? Base analogues. The chemicals which have analogous structures to the base, those 4 bases are called base analogues. So they are similar to the bases of DNA, that is purines and pyrimidines, or structurally resemble the DNA bases. And best known base analogues are bromouracil, 5-BU, 5-bromouracil, aminopurine, which are incorporated into DNA instead of normal bases during the process of replication. The 5-BU, 5-bromouracil, they can be artificially synthesized and they can be used in genetic research. They can be incorporated in DNA in the place of thymine and to know the effects. Okay. Then, instead of the methyl group of thymine, the bromouracil contains bromine group, Br group, which highly resembles the thymine. This pairs with adenine, as like the thymine, and brings about mutation. So, what I have shown here is that 5-bromouracil bonding with adenine. Okay. Then, here you can see the incorporation of 5-bromouracil, and when replication takes place, so Bu gets incorporated and therefore bringing about mutation. Then chemicals changing the specificity of hydrogen bonding. There are many chemicals which do this, okay, like nitrous oxide, HNO2, hydroxylamine, HA, then EMS, ethyl methane sulfonate. Okay, they bring about change in the specificity of hydrogen bonding, like double bonding and triple hydrogen bonding, like that. Then what does this nitrous oxide do? It converts the amino group of bases into keto group through oxidative deamination. The order of frequency of deamination, that is removal of amino group, is adenine greater than cytosine greater than guanine. Then deamination of adenine results in formation of hypoxanthine, the pairing behavior of which is like guanine. Hence it pairs with cytosine instead of thymine, replacing AT pairing by GC pairing. Okay. Then deamination of cytosine. Deamination of cytosine results in the formation of uracil by replacing amino group with hydroxyl group. The affinity of hydrogen bonding of uracil is like thymine. Therefore, CG pairing is replaced by UA pairing. Okay. Then, here you can see deamination by nitrous oxide of adenine into hypoxanthine as depicted in diagram A, and cytosine into uracil as depicted in the diagram B. Okay, then. So NH2OH, that is hydroxylamine, it hydroxylates with C4 nitrogen of cytosine and converts it into a modified base via deamination, which causes two base pairs like thymine. Therefore, GC pairs are changed into AT pairs. So, next category of chemical mutagens happen to be alkylating agents. Addition of an alkyl group to the hydrogen bonding, oxidation of guanine at N7 position and adenine at N3 position, residues of DNA is done by alkylating agent. As a result of alkylation, possibility of ionization is increased with the introduction of pairing errors. Hydrolysis of linkage of base sugar occurs, resulting in gap in one chain. So this phenomenon of loss of alkylated base from the DNA molecule is called depurination. So depurination is not always mutagenic. The gap created by loss of a purine can effectively be repaired. Then these are some of the alkylating agents: DMS, dimethyl sulfate, then EMS, ethyl methane sulfate, then EES, ethyl ethane sulfate. Then next category of chemical mutagens happen to be intercalating agents. Ethidium bromide ETBr, which is used in agarose gel electrophoresis age, is one of the intercalating agent. See what happens here is, and some of the other intercalating agents like proflavin, acridin orange, they also operate in the same mechanism of ETBr. What they do is, these molecules intercalate between the bases of DNA and disrupt its structure. So you can see that there are normal DNA molecule, and when you add acridin, you can see what happens, right? And as a result, there is deletion, and it will block transcription also. Then other chemical mutagens which bring about mutations are metal ions. Then ROS. ROS is reactive oxidation species. Then benzene, synthetic rubber and rubber products. Then sodium azide, aromatic amines, alkaloids and deaminating agents. So after having studied about physical mutagens and chemical mutagens in detail, now it is time for us to talk about biological mutagens like viruses, bacteria and transposon. So viruses are the common mutagens which are known to us, and they cause lethal health issues. Viruses insert their DNA into our genome and disrupt the normal functioning of DNA or a gene. Once it inserts the DNA, that DNA is replicated, transcribed and translated, and viral proteins are produced instead of our own proteins. This is how they bring about mutation. Best example, human papilloma virus, HPV virus, that causes cervical cancer, uterine cervical cancer. Then bacteria. Some of the bacteria are also dangerous for our DNA. They cause inflammation. It provokes DNA damage and DNA breakage. Helicobacter pylori, that is transmitted through contaminated food, is responsible for the formation of ulcers in the alimentary canal. Then transposons, popularly called jumping genes. In fact, they are less known biological mutagens. They are non-coding DNA sequences, jump from one place to another place in a genome, hence called jumping genes, and influence the functioning of the gene. And these are our own DNA. They are not foreign DNAs, believed to be originated from the retroviruses. Then, dear students, These are the list of references which madam would have referred to make these informative PPTs. Now it is the time for us to discuss some of the assessment questions. First MCQ is example of a physical mutagen: UV radiation, X-rays, gamma rays, D all the above. Invariably answer is A, UV radiation. Second question, 5-BU, 5-bromouracil is the analog of which base? A thymine, B guanine, C cytosine, D uracil. Answer is A, thymine. Please note, 5-bromouracil is the analog of thymine. This substitution allows the base to mispair with guanine via enol tautomer. Okay. Then question number 3. Which of the following is an example of intercalating agent? A ETBR, B 5-BU, C purine, D clastrogen. Answer is A, that is ETBR, ethidium bromide. Question number 4. Which base is generated by deamination of 5-methylcytosine? Thymine, B adenine, C guanine, D uracil. Answer is A, that is thymine. Please note, deamination of 5-methylcytosine generates thymine. This thymine pairs with the thymine in the next round of replication. Okay. The next question, the class of mutagens responsible for chromosomal breakage, deletion, duplication and rearrangements are teratogens, carcinogens, clastrogens, intercalating agents. Answer is C, that is clastrogens. Okay. My dear students, it is the time for me to end my talk on mutagens. Thank you so much for your patient hearing. So far we have discussed about different types of mutations in our previous session, and in this session we have discussed in detail about the different types of mutagens which are responsible for bringing about mutation. Thank you for your patient hearing. Let me wind off my class today.